I changed my title to Impact of Dengue Cross-Reactive Antibodies on Zika virus infection and seeding of the placenta. It's always a hard act to follow after Maro, and I certainly appreciate the conference organizers for giving me the opportunity to present some of our work today. So my laboratory is broadly interested in flavivirus infection. We've been interested for quite some time. This is my no conflict of interest. So we've all heard about Zika virus, but a couple of things that I'll point out in this case, and I think we were just having a nice discussion about ADE, which I think transitions very nicely into the presentation that I'll have today, about the relationship between dengue and Zika virus. You can clearly see that on a slide like this, that Zika virus is closely related to dengue virus as well as some other flaviviruses. The protein that we'll be discussing today is the envelope protein, which is found on the outside part of Zika virus. And again, my laboratory is broadly interested in understanding aspects of virus-host interactions with flaviviruses, in particular Zika and West Nile and dengue. And we've done quite a bit of work with Zika virus at the molecular level, trying to understand aspects of how the virus antagonizes the innate immune response. And we made some very interesting discoveries. First is that the virus seems to block interferon beta production. People typically measure interferon beta by qPCR. What we find is that Zika virus doesn't seem to block the ability of the cell to induce interferon beta at the mRNA. There seems to be a specific blockade of either interferon beta at the translation level or the secretion level, and this is something that we're studying in quite a bit of detail. Secondly, we've found that the virus blocks STAT1 and STAT2 phosphorylation. Adolfo Garcia-Sastria's group has shown that STAT2 is degraded. In addition to that, we've shown that the phosphorylation events of these two specific transcription factors are also blocked. In work that's unpublished, we've now identified STAT5 as an important transcriptional regulator of dendritic cell activation. And it seems as though Zika virus, along with West Nile virus, seem to block this. What's interesting is that dengue virus doesn't seem to block this. So there seems to be some specificity amongst these flaviviruses in terms of their ability to antagonize the innate immune response. But today I wanted to focus specifically on the role of dengue cross-reactive antibodies and how they play a role in Zika virus seeding of the placenta. So one of the major questions that my lab wanted to better understand is how this whole aspect of vertical transmission of Zika virus from the infected pregnant mother to the developing fetus. And we can certainly see this here, resulting in microcephaly and fetal demise, most certainly. And one of the things that my laboratory first discovered was we identified that Hofbauer cells, which were discovered over 100 years ago, are found within the placenta, within the villus stromas, are tropic cells for Zika virus. These seem to be very permissive for Zika virus infection. We've shown that they can replicate. We don't see a lot of cell death. And just like I mentioned before, we don't see any type 1 interferon secretion, and we see very minimal cellular activation, which corresponds to our STAT5 inhibition. This work was followed up on by fantastic work from Sharif Zaki's group at the CDC. And there's a couple of things I wanted to note from his studies. First and foremost is that Zika virus persists not only within the fetal brain, but most importantly, I think what tends to get ignored is it persists quite often in the placenta. Okay, so from mother onset of symptoms to when Zika virus is detected within the placenta, they showed across several individuals that Zika virus can persist whether the mother is infected in the first, second, or third trimester, and it can persist for anywhere from 19 days to 238 days post-mother onset of symptoms, and then it can eventually go to the brain around a mean average time of about 163 days. On a follow-up study, what they found was that Zika, approximately 50% of pregnant mothers that they analyzed had Zika virus RNA within the placenta. So there's quite high amount of individuals that seem to have Zika virus RNA within their placenta. Within these placenta, what they found from a couple of individuals was they looked at in situ hybridization both for plus strand and minus strand Zika virus RNA, and they found that Zika virus RNA does localize within the placenta, and most importantly, they localize within the Hofbauer cells that we had identified. They don't really see much replication in other cell types within the placenta. The Hofbauer cells seem to be the primary cell that's permissive for Zika virus replication within the placenta. So we really wanted to better understand, now that we've identified what cell types are infected within the placenta, how exactly does the virus cross this placental barrier, which seems to be quite a robust barrier for the virus, for any virus to cross. We did some work with Jens Rammert's group at Emory University, looking at a panel of monoclonal antibodies that the lab had generated against dengue virus, a panel of 48 monoclonal antibodies. We screened them against Zika virus and found a variety of responses. First, we identified monoclonal antibodies that can bind to whole Zika virus lysate, can bind to Zika virus itself, the virion, but very few of them actually neutralized the virus when we performed a focus forming assay. Whereas in the context of dengue virus, we can see many of these monoclonal antibodies can actually block dengue virus infection. So what we did was we took 4 of these antibodies that vary in their ability to neutralize as well as bind the virus, and that's shown here. We included one that was a high neutralizer. This is based on studies done on viral cells, which again don't express FC receptors. We have one that's more of a weaker neutralizing antibody, and one that can bind but can't neutralize, and one that doesn't bind nor does it neutralize, and we included a non-specific control. So what we can do in our laboratory is we can take term placenta, we can isolate these Hofbauer cells, we then generate immune complexes with Zika virus and antibody. We combine these with our Hofbauer cells, we wash and incubate, and then analyze virus replication within these Hofbauer cells in a variety of different methods. First and foremost, we can look at E protein staining to look at the frequency of infected cells, and what we can show is that we can go from about 5 to 10% infection all the way up to about 90%, 80 to 90% infection. One of the remarkable things about this data was that it didn't matter whether we had a neutralizing, poor neutralizer, or one that didn't neutralize. As long as the antibody bound the virus, we seem to get this enhancement of infection. The other striking thing that I still don't have a good answer for, but probably requires a lot more work in terms of the mechanisms of antibody-dependent enhancement of a cell, is why we didn't see this classic neutralization at very high doses, followed by enhancement, followed by low levels of infection. Rather, what we see is high level of infection at any dose that we see past 0.4 micrograms per mil. We also show that we can get infectious virus release from these cells as well. We've also gone on to do a number of different studies, and this work has recently been published. We show that there's minimal effects on activation. We see minimal effects on cell death 48 hours post-infection. And most importantly, similar to what other people have shown, is that these immune complexes do increase the amount of viral binding and entry to these Hofbauer cells. So we wanted to next ask, which has not been really looked at for dengue virus or any other pathogen, is what about other IgG subclasses? So the antibody that I've shown you so far is based on an IgG1 FC. With the help of Jens Rammert's lab, we looked more closely at the various different subclasses of antibodies. The first thing that we did was we looked in our Hofbauer cells from term, and we've also done second trimester as well, and we see very similar profiles of what the Fc gamma receptor expression is on these Hofbauer cells. These Hofbauer cells seem to express high levels of Fc gamma receptor 2, and to some degree Fc gamma receptor 1, but very little Fc gamma receptor 3 is expressed on these Hofbauer cells. So we next wanted to ask what effect does this have on other subclasses of IgG, enhancing Zika virus infection in these Hofbauer cells. So again, with the help of Jens Rammert's lab, we generated various subclasses of antibodies that have the same exact epitope region where this antibody binds to the virus. We also created a LALA mutant of the IgG1 version of this as well. And what we find is that in a dose-dependent manner, we see differences in IgG subclass dependent enhancement of our Hofbauer cells. What's interesting is that we see IgG1 and IgG3 showing the highest levels of antibody-dependent enhancement. What's interesting is IgG2 is a very poor enhancement ability within these Hofbauer cells, suggesting that this may be operating through an Fc gamma receptor 2, possibly one dependent mechanism for entry. And this is something that we're looking at in a bit more detail. But what this does show is that the antibody-dependent enhancement is IgG subclass dependent. We next wanted to move our studies into more relevant model systems. So rather than just simply isolating these cells from human placenta, we next wanted to look specifically in human explants. So what we were able to do in the laboratory, we can isolate these chorionic villi that maintain the architecture that's normally found within the placenta. So we are able to obtain first and second trimester placenta. We then delicately isolate these chorionic villi, which are again normally bathed in mother's blood, which is shown here, but still maintain all the architecture of the syncytial layer. The cytotrophoblast layer have endothelial cells, have fibroblasts, have our Hofbauer cells, all intact within this system. And what we did was we took these placental explants, we generated our immune complexes, we treated them with Zika virus or with these immune complexes, washed and incubated, and see if we can detect virus replication. And much to our surprise, we now see Zika virus replication within these placental explants in a dose-dependent manner, as well as in an IgG subclass-dependent manner, very similar to what we saw in the context of our Hofbauer cells. What we find, we can look at both plus-strand virus RNA as well as minus-strand, which is that replication intermediate. And what we find in a dose-dependent manner, IgG1 and IgG3 seem to have the highest level of placental seeding when we use these immune complexes, whereas IgG2 seems to be much more poorer in its ability to actually seed the placenta. And again, just throwing on Zika virus alone, we see very little replication within these placental explants, which is similar to what either Mike Diamond or Carolyn Coyne or Jonathan Miner have shown in their systems as well. We next wanted to determine whether when we put on these immune complexes, what cells are they actually targeting? Are we seeing the similar findings that the CDC had from actual patient data? And what we find in this context is that when we throw on these immune complexes, we actually find these Hofbauer cells are the primary cells that are infected. We can see this by Zika virus antigen staining here for the viral E protein. And this corresponds to CD163 staining, which is another marker for our Hofbauer cells, these placental macrophages. So what's fascinating is that we can now take these antibodies, throw them onto the placental explants. We can now identify these Hofbauer cells are infected. We wanted to better understand transplacental IgG transport within these samples. We wanted to determine what role FcRn, if any, plays, the neonatal Fc receptor plays in transporting Zika virus across using these immune complexes. We wanted to study this, but we decided to use this little trick of using protein A, which is seen here, which was discovered many years ago to bind to the same exact place on IgG1 as FcRn normally binds. Okay, so you can outcompete FcRn binding on IgG1. But what's interesting is that IgG3, protein A doesn't bind to the same region. Actually, I don't think it binds IgG3 Fc at all. So you're able to determine specificity and determine what role FcRn, neonatal Fc receptor, plays in transporting these immune complexes across the placental barrier. And what we find in a dose-dependent manner when we throw on these immune complexes with increasing amounts of protein A, and this is a fold decrease graph, we see a significant reduction in the amount of virus replication occurring within these placental explants. If we do the same experiment in the context of IgG3 generated immune complex, which showed similar levels of virus replication as that of IgG1 immune complexes, we see absolutely no effect. We've also gone on to show using FcRn blocking antibodies, we see the same exact effect as well. So this suggests that the transport is FcRn dependent. We next wanted to ask what happens in the context of a LALA mutant IgG1 antibody, and what we find here is a significant reduction when we throw on immune complexes using the IgG1 LALA mutant, demonstrating that the transport of Zika virus across the placental barrier is not only occurring in a neonatal Fc receptor dependent manner, but that this enhanced infection occurs within the villostroma in an Fc dependent manner. I think that's just a byproduct at the end of the day of these immune complexes transporting the virus across. And so in a normal situation, if the virus somehow managed to get across, you may only infect a very small percentage of Hofbauer cells within the villostroma. These immune complexes not only allow the virus to get across this placental barrier, but now it just increases the likelihood of seeding that placenta, so many more Hofbauer cells may get infected. We next wanted to ask what about some of the host response that's found within the placenta. So Carolyn Coyne has done some beautiful work looking at the syncytial trophoblast layer and shown that this placental layer secretes high levels of type 3 interferons normally. And this is a mechanism that the placenta uses to prevent Zika virus and several other viral infections within this layer. And so we wanted to ask, what effect does type 3 or type 1 interferons have on blocking Zika virus infection within these Hofbauer cells? Again, these placental exocytes normally secrete type 3 interferons, so if we're throwing these immune complexes, is it blocking the ability of Zika virus to infect these Hofbauer cells? And much to our surprise, what we find is that interferon lambda has absolutely no effect on the Zika virus replication within our Hofbauer cells, despite that syncytial trophoblast layer secreting high levels of interferon. Rather, interferon beta has a surprising, not surprising, but it has a strong effect on Zika virus replication. It can certainly reduce the amount of virus replication occurring within these cells. And it occurs either without antibody-dependent enhancement effects or with antibody-dependent enhancement. We next wanted to look at whether or not this virus can induce type 1 interferons, similar to what we had seen in either without Zika virus enhancing effects in Hofbauer cells or within our human dendritic cell work. And what we find is that at the mRNA level, we see high levels of transcription occurring between interferon, type 1 interferons, type 3 interferon, either alone without enhancing antibody in the presence of our neutralizing antibody or in the context of our immune complexes. But what's fascinating is that we see absolutely no production of type 1 interferon at the protein level. If you measure this by ELISA or Luminex, we see absolutely no detection. Normally, Hofbauer cells, I guess when you isolate them and throw them into culture, you do get some level of interferon alpha secretion occurring. But what's interesting, as you increase the amount of infection using our antibody-dependent enhancement conditions, we see reduced amounts of interferon alpha being produced. And we see absolutely no interferon lambda being produced by these Hofbauer cells. In data that I'm not showing, we've also treated these cells with TLR agonists, TLR2, 3, 4, 5, 7, 9, as well as RIG-I, MD5. We treated them with STING and CGAS ligands. They all seem to induce cytokines within these Hofbauer cells. But somehow when Zika virus infects them, it has a way to completely block the amount of protein that's secreted from cytokine levels within these cells. So this may be a mechanism by which not only the virus can block JAK-STAT signaling, it can also block the ability to induce a pro-inflammatory cytokine response, and that probably leads to why this virus goes persistent within the placenta without inducing overt levels of inflammation, which would be detrimental for fetal development. And so with that, I'll leave you with the summary slide. I think we've been able to show that cross-reactive dengue antibodies can enhance Zika virus infection within these Hofbauer cells, but most importantly, that these immune complexes can help transport the virus across that syncytial trophoblast layer, leading to a greater seeding of these Hofbauer cells. Type 1 interferon, but not type 3 interferon, can restrict Zika virus infection of these Hofbauer cells. We can see this sort of in these blue triangles that are shown here. This is all the interferon lambda that's secreted by these syncytial trophoblasts. That is not able to stop the virus from replicating within these Hofbauer cells. And that Zika virus infection severely diminishes type 1 and type 3 interferon production. With that, I'll acknowledge the folks in my laboratory. This work was led by an MD PhD student in my laboratory, Matt Zimmerman, as well as a former graduate student, Kendra Quick, who's now in Greg Ebel's lab, and Aaron Burrell's lab, as well as other folks in my laboratory, collaborative work with Jens Rammert's lab, as well as Rana Chakraborty. We have a UO1 grant to study these Hofbauer cells in much more detail. We had some help with our histology by Deepa Makaya, as well as work that we have ongoing with Carolyn Coyne and Nitin Arora, who now has his own lab at University of Alabama, Augustin Rajakumar, Pediatrics Flow Core, and all of our funding sources. With that, I'll take any questions that folks may have.